Good morning, everyone, and welcome to the Weight Research Institute webinar series. This morning, I welcome Pietro Previtali, who is a PhD student of Christopher Ford. Pietro's almost finished his PhD and returns to Italy next week. And the title of his talk is Studying Unknown, Undefined, Unresolved Sugar Flavour Nexus of Grapes. Over to you, Pietro.
Thanks, Rebecca, for the kind of introduction. And thanks, people, for just joining in. This will be a few research highlights about this project, about the sugar flavor nexus. And I want to start thanking my supervisors, Christopher Ford and Kira Wilkinson. And because this project is part of the Training Center for Innovative Wine Production, this project is affiliated with an industry partner, that is EJ Gallo Winery, and my supervisor there is the mighty Nick Dukuslian. So brief, let's see if it's going.
Just give it a second.
Yeah. Just outline of the talk today, introduction, background information, and the definition of research questions. Then we'll head into the main research component. This will be structured in 3 different blocks, reflecting 3 approaches to study the sugar flavor nexus: single berry, some field trials, and the meta-analysis approach. And then some conclusions and future perspectives. So why should we bother studying the sugar flavor nexus? Well, because we know that these components are key quality traits for wine grapes. And to understand the relevance of these components on the sensory profile of wine, we could all think about a wine without ethanol, without alcohol, or a wine with too much alcohol, or again, a wine without aroma compounds. And I had that experience myself when I broke my nose in a very hilarious way. I'm not going to tell you how that happened, but you smell the wine, you cannot perceive anything, and then you taste it, and all you get is just the basic taste. So it's pretty important to understand the relationship between sugars and aroma compounds to drive production of grapes and to meet specific quality targets. It's particularly urgent to look at it right now because anecdotal observations have seen that grapes at harvest are displaying higher sugars and lower aroma compounds. Before we get into the sugar flavor nexus, I thought it was important to look at these components in grapes individually, so separately for sugars and aroma compounds. We know quite a bit about sugars in grapes. This is related to a bulk of work done by researchers such as Brian Coombe on this campus and Mark Cleaver in UC Davis and many other collaborators. The physiology of carbon assimilation in grapevine is not different from any other plant. So you can see in that panel A, we have this complex chain of reactions that are needed to harvest light and convert that physical energy into chemical energy to assimilate sugar. Once sugar is assimilated, fluxes of carbon between grapevine organs are called the carbon partitioning dynamics. And we know that in grapes and grapevines, these are ruled by the source to sink relationship. So source organs are those who make photosynthase, and sink organs are the ones demanding for photosynthase. And in panel B, we can see that at different stages of the development, the very same organ can display sink or source behavior. Certainly, the most important thing and aspect about sugar accumulation when we look at the sugar flavor nexus is the carbon and water balance in the grape period. And one could think that changes in changes in sugar content are the only thing that matter to explain sugar accumulation. However, from a winemaking perspective, we are interested in a concentration of sugars, and that's related to amount of water or the weight of the berries. And this is why it's really important to also account for changes in berry weight, which are described by the amount of water in the berry. So again, there's a lot of literature on this, and I've tried to summarize it, the carbon and water balance in the berry in this panel A, and then I've reported some key measurement of key processes into panels B to E. So we have water getting into the berry, either via the xylem or the phloem, depending on the stage of development. And then berry can leave, water can leave the berry either via transpiration through the cuticle or via xylem backflow. So that water is going back to the plant to be redistributed to other organs. On the other side, we have sugar getting into the berry via the phloem, and then some of it is respired, so it's used to sustain growth or development processes. So on one side, we have water accounting for the fresh mass of the berry, and on the other side, we can look at carbohydrates as the main component of dry mass of the berry. So in panel B, we can see, are they going to be able to see it? I'm just going to do this, sorry. In panel C, we can see panel B, we can see the changes in fresh and dry mass in the berries. So these are changes in berry weight and display a double sigmoid curve. The accumulation of carbohydrates in the berry is actually happening a bit later, normally starting after 50 to 60 days after flowering. And that is the process of sugar accumulation, which starts to happen at about veraison, when the berries start to get colored and soften. In panel C, we have the changes in carbon imported and respired. So again, we can see the starting at about veraison because of a strong sugar accumulation happens in the berry. We have a very steady rate of carbon import into the berry. We can also see that during the ripening stage, even though we're having quite a bit of carbon imported into the berry, just a tiny bit, a tiny fraction of it is actually being metabolized again. And then when we look at panel D and E, we have changes of berry transpiration, phloem flow, and xylem flow. And again, this can be negative at different stages of development. When we think, we look at aroma compounds in grapes, this is going to be tricky. These belong to, actually, sorry for the training center, I'm just going to put it down. Maybe here. These aroma compounds belong to classes of volatile organic compounds of box found in all plants. You can see the most important ones for grapes and the attributes, sensory attributes in wine. These compounds, aroma compounds, from a wine perspective, those aroma compounds derived from grapes are called as varietal aromas because they're strongly dependent on the variety. And in general, as opposed to sugars, which we would normally consider primary metabolites, aroma compounds are secondary metabolites because they're more involved into protection of the plant against biotic and abiotic stresses, and also they support the reproduction. So they are subjected to genotypic and environmental regulation. And an aspect to be very mindful of is that aroma compounds can be present both as free volatile compounds or glycosylated and therefore bound to sugar moieties. And when they are glycosylated, they're not volatile. So what about the sugar flavor nexus? What about a potential relationship between these sugars and flavors? There have been sparse attempts to define this relationship in literature. In the beginning, I started looking into literature about the interconnection between primary and secondary metabolites. However, I keep thinking back at a meeting with Chris when he pointed out that because we're seeing so much sugar getting into the berry and not being remetabolized, sugars in grapes during the ripening stage actually display more a behavior that is similar to secondary metabolites rather than primary. So there have been some attempts to define switches in relationship between primary and secondary metabolites at the gene expression or transcriptional level, but there's not enough literature in terms of sugar and flavors just there. The most bulk of information about this sugar flavor nexus is found when looking in studies on the evolution of aroma compounds during berry development. This started back with Kumba McCarthy. They tried to define a common nomenclature for the process of development of aroma compounds in grapes, and they called it engustment, which means development of aromas. To do so, they did some berry sensory tasting at different Brix levels, at different sugar levels, but also they measured this red-free glycosyl glucose, which is a proxy for the concentration of glycosides of aroma compounds. It's red-free because it doesn't account for glycosides of phenolics. What they saw is that there's actually a very sharp increase in this red-free glycosyl glucose starting after 20 Brix, and they also saw very more fruity notes into the berry starting at this point. And so they said that the inception of aroma development in grapes starts at 20 Brix, or just after that. Following studies have supported this hypothesis by looking at the volatile organic compounds of grapes at sequential stages of development. So pretty much they were able to report chemical concentrations of different aroma compounds responsible for different attributes from flowering to harvest or just during the ripening stage. And again, there's quite a bit of variation in these studies and in what analysts they targeted and what varieties they looked at. But the general understanding is that throughout ripening, there's a decrease in compounds that are responsible for green notes. These are C6 compounds and IBMP. And around harvest, again, late, just at about or after 20 Brix, there's an increase of fruity compounds, mostly in their glycosidically bound form. The final bit about literature about the sugar flavor nexus is found in studies that have tried to establish a link between grape maturity and wine style. And I'm referring to wine style in a very generic style of wine, red fruit, dark fruit, dried fruit. And this is work done by Alain Denoir and colleagues at CSU. They developed a model called the berry aromatic sequence. So they related wine styles to changes in sugar per berry. So here we're talking about content of sugar. Obviously, the content starts increasing from variation to a certain time, which we could call a plateau of sugar accumulation. And this is due to the fact that phloem input of sugar decreases and then finally stops. And they have created some predictive models saying that it is possible to harvest grapes at a certain number of days after this plateau day to target a red fruit rather than or dark fruit or a dried fruit. And this is variety specific. So this is the case for Shiraz and Wagga Wagga. So a little bit of literature is out there for the sugar flavor nexus, but still plenty of stuff and work to do. We developed 3 research questions. The first one is there a relationship between sugars and aroma compounds? And we wanted to more accurately and directly look at the metabolic level and look at this relationship between sugars and specific aroma compounds of interest. The second one was what is the effect of sugar accumulation rates on flavor development? And this and here the idea was if the 2 processes are tightly linked, when we manipulate one, we should see a similar effect on the other one. Whereas if we're able to change one without having any effect on the other ones, probably that's going to tell us that there's not really a strong relationship. And finally, and this is the bit that is mostly related and relevant to industry, what vineyard practices are most suitable for the production of grapes with less sugar and more flavor? So to answer the first question, we thought that we needed to probably move towards a single berry approach. And this is because the main limitation of previous studies were 2. The first one was that these studies used developmental or chronological scales as a proxy of sugar rather than sugar measurements directly. So I've attempted to summarize 7 different studies normalizing their concentration in C6 aldehydes, C6 alcohols, and C6 acetate esters and reporting them using the same scale, days post variation. So different points, colors, and shapes are different studies out there. And we can see that there's quite a nice trend for some compounds, but still a large dispersion. And this is due to the fact that they use different varietal grape varieties, also different experimental conditions. They analyze they target the sequential stages in very different ways. They also reported for their sequential stages of development, the TSS levels. So I've converted this trend into the TSS in bricks trend. And we can see that again, we can see some nice trends, but there's still a lot of variation. And it sort of looks that these studies have targeted main or key stages of development, whereas in certain bits, we have a few points there. So this is certainly not the answer. It's a pretty nice approach to summarize literature, and we need a more direct measurement of aroma compounds and sugars to understand their relationship. Also, these studies have used mixed berry populations, meaning that they mixed up berries present in the vineyard at a specific sampling date. And this is good to describe changes, for instance, from an industry perspective, but at the same time, it doesn't account for the asynchronous development of grapes. And this, in fact, was advocated by Kumba a while ago, that a single berry approach needs to be used when looking at metabolic changes due to the stages of development. So we decided to use a single berry approach. This is an example of the asynchronicity of berry ripening here at the Alderstoke Vineyard, and the whole study is published in the Yellow Journal. So we started off with a sequential target sampling, went out to the vineyard over 9 weeks on a weekly basis, and picked single berries at different stages of development. The 1 to the 4 were green hard berries, green soft, pink blush, purple, and blue berries. These were discriminated by color and touch characteristics. And then blueberries were further separated by bricks, and shriveled berries were further discriminated by visual characteristics, so the presence of shriveling symptoms, and SH1 to SH2 were separated by bricks. So we know we can use this discrimination protocol because there's a pretty strong correlation between the classes of development and the bricks. So this means that whenever we pick a green berries, either today or in 3 weeks, those green hard berries, their TSS is going to fall within the same range. We cannot say the same thing about berry fresh mass. It's much more variable and it's no news. But the point of showing this chart is that the range in average fresh mass by class was between 0.3 and 1 gram. So we really had very little amounts of samples for this analysis. So the way we did this was to actually get a single berry, weigh it, and record TSS as a proxy for sugar on half of the berry, because that doesn't need very much juice. The rest of the berry was used for sample preparation for volatile analysis. We adopted a method from ARRI staff, and we were able to scale it down to 0.3 grams of powder. This allowed us to measure six compounds as a starting point in about 150 single berries. So for the first time, we were able to look into the lipoxygenase pathway. So that one that is leading to the conversion of C6-aldehydes into C6 alcohols and their isomers. We were able to look into these metabolites and report the relationship between their concentration in micrograms per gram of berry and berry TSS. So for each metabolite here, we can see each point is a single berry and the color is assigned according to the classes of development. So we saw that, I'm taking here as an example, E2-axanol and XM1O. We saw that not only berries belonging to the same class of development, pretty much the same color, are grouped together in terms of their TSS level, these are sugar levels, but also in terms of that metabolite content. And this means that the stage of development is a main driver for the concentration of these compounds. We took it a step further and we approximated the content of this compound. And this way we were able to actually to be more confident that the increase we saw up to 20 to 24 bricks and the decrease thereafter were active changes in the berry, and they were not driven by concentration or dilution effects because of differences in water content. So just a summary about the single berry approach. This we targeted directly changes in C6 compounds and sugars in single berries, and this allowed to prevent the effect of asynchronous development, and this needs clearly to be extended to other grape volatile compounds that may or maybe not dependent on developmental stages, but still this will need some method development to happen. The second approach was some field trials about ripening manipulation. Because of climate change, in particular elevated temperature and water stress, we know that sugar accumulation is accelerated in our vineyards in most of the regions worldwide, and this has negative effects on the grape composition. This has been described as the coupling between sugars and organic acids and sugars and anthocyanins. And this is pretty clear in studies that have looked at the TSS to organic acids or TSS to anthocyanins ratio in vines submitted to heat treatment or water stress treatment or a combination of the two. However, there is little literature or no literature on the effects on aroma development, and specifically in grapes. There's a little bit of work on the effects on the berry sensory and the wine sensory, wine metabolites, but not really much about aroma development in grapes. So we targeted this subject with some vineyard trials in California in 2019. We did the same trial on Riesling and Cabernet Sauvignon in 2 different regions. And the outline of the study was to use, the idea of the study was to use crop load and late season irrigation, so irrigation ramped up late in the season, apply the treatments to the vines to create different rates of sugar accumulation, to then pick grapes at the same TSS level, but obviously reached on a different date, and characterize the effects on the global composition of grapes in fast, control, and slow grapes. And this is work that has been recently published in the JAG Food Chemical Journal. So you're looking now at the sugar accumulation curves for Riesling. We started this late season irrigation, meaning 50% more irrigation at 17 bricks, this red dotted line, and we harvested grapes at 21 bricks, just about here, which was the commercial maturity. Here we can see the 4 treatments, and the abbreviation could be a control in blue, thinning as in cluster thinned in green, late season irrigation only in red, and the thinning plus late season irrigation in yellow. So we can see that in Riesling, the only separation in terms of sugar accumulation dynamics was between low crop load and high crop load, and obviously low crop load, and this is pretty established, accelerated ripening there. But there was no effect of the addition of extra water in either low and high crop load. Picture is quite different for the Cabernet Sauvignon. So same treatments here, but we can see that in this very messy chart, I should say that we started late season irrigation at 20 bricks, and we picked the grapes at 26 bricks. And again, this was the commercial standard. The most important thing here is that we were able to harvest grapes at the same bricks level, 26 bricks, within the same vineyard at 3 different dates, and the time spent between the earliest and the latest was 3 weeks. Here we can see the sugar accumulations between 20 and 26 bricks split by crop level. So in low crop load, we achieved the fastest sugar accumulation just by thinning the vines, and when we applied extra late season irrigation, we had these three weeks delay, which was then leading to this lowest sugar accumulation. In high crop load, we had our control, the blue line, but the application of late season irrigation didn't really cause any delay. So there is an interaction between crop load and the way that water is being uptaken and its efficacy in the late ripening. We looked into vine measurements and specifically the vine water status and assimilation patterns. And we were able to see that actually to explain this slower sugar accumulation by the fact that these berries were actually uptaking more water and replacing berry transpiration. In fact, we know that after 20 bricks, any increment in sugar is related to berry dehydration. It's not an active sugar accumulation, and therefore those grapes were actually replacing the water, decreasing the increments of sugar. So we wanted to understand the effect on the local chemistry, and particularly we wanted to understand what factors are driving the concentration of a bunch of compounds. Free aroma compounds, bound aroma compounds, phenolic compounds, and basic chemistry. To do this, we used hierarchical clustering analysis on the concentration of compounds normalized by compounds across the treatments. So in these heat maps, you can see the normalized concentration of each compound. The compounds are in rows, and or wine or grape parameters, are in rows, whereas we have treatments listed from fast to slow in columns. And the best clustering solution to describe trends of variation across these treatments was 5 clusters. So we saw that some compounds are strongly dependent on the ripening rate. So here I've listed the treatments from fast to slow, and we can see that for some compounds, the concentration is highly increased in slow ripening. You can see this going up at the end. These are important phenolic compounds, but also nitrogen components. Some other compounds were decreased in response to the ripening rate. So the later we pick, and the lower concentration of these compounds. We have a bunch of aroma compounds in their free form, and these are expected to be volatile and lost by the berry. We think this is due to plant signaling processes, but also no news that we had a decrease in titratable acidity, tartaric acid, and moisture. Some of the compounds where their concentration was a bit more irrespective of the ripening rate, and was mainly driven by crop load conditions. So total phenolics and a very important fruity compound, also considered a fruity enhancer in its bound form that can be released later in the wine, was present at higher concentration in low crop load, whereas some of the aroma compounds and bound aroma free and bound fractions were increased in high crop load. So in general, we really saw that a slower ripening has a positive effect on the grape composition because it's leading to less grain compounds and more fruity compounds. And we characterize this at the chemical level. We also went to the wine chemistry, so did some fermentations of 65 kilos fruit lots. And here I've reported a PCA of the treatment, control fast ripening and slow ripening, and there's a pretty nice separation. We can see that the control was most associated with this grassy compound and this flora compound. But when we went to the slower sugar accumulation, this was associated with increased concentration of beta-damascenone and linalool, grape-derived fruity compounds, and some fruity esters, but also higher levels of color and phenolic compounds. So second approach, field trials on ripening rate. We aim to create a model system to explore effects of different ripening rates on grape and wine chemistry. We saw different outcomes in Riesling and Cabernet Sauvignon. These are explained by different targeted maturity for stylistic preference. In fact, we're picking Riesling at 21, which is not long after the 20 bricks, and therefore we are not into the passive dehydration process of the grapes. And we actually saw that for that reason, when we added extra water, 17 bricks, we actually caused a bit of an increase into assimilation and sugar accumulation. So in general, we saw that lower sugar accumulation is important to improve grape ripening, especially in red varieties. This is because of less green and more fructifloral aroma compounds. And we had a media release about this very specific outcome. This has started a very nice conversation with industry about it. And industry has been very interested in this outcome. The third approach is it was a bit of a new thing for me, and it's the meta-analysis or more comprehensive analysis. So because we saw that delayed ripening, the lane ripening, and slower sugar accumulation is so positive for grape composition, we wanted to do a quantitative review of vineyard practices. There have been qualitative reviews, but no quantitative reviews, so we wanted to focus on data-driven analysis. And this is where meta-analysis jumps in as a pretty cool tool. So meta-analysis is pretty much based on gathering studies on a specific topic, extracting quantitative data from them, and then analyze them with specific statistical tools to draw new conclusions. This will be a brief outline of the work and some relevant findings. So the first thing is the definition of a quantitative variable of interest. For us, it was a measure of ripening delay, and this could have been either a number of days between when you pick, control, and treated grapes at the same sugar level, but that doesn't have any measure of the precision, no standard error, no standard deviation. So we actually decided to characterize the ripening delays as a difference between the TSS and the control and the treated on the very same day. So this is pretty much a model of how it works. So we pretty much looked at sugar accumulation curves and extracted this delta TSS. Obviously 1, 2, 3 bricks, this can be easily translated into a quantitative scale of ripenings. We did this on a wide number of studies, and we applied some important inclusion and exclusion criteria. So we ended up with 35 studies, but because within each study, any comparison is a single individual comparison, and studies can report multiple comparisons for varieties, blocks, sites, different treatment intensities. We ended up with 204 actually comparisons, which is pretty good because this is a good number to treat statistically. So meta-analysis was composed of a preliminary analysis on all the techniques to delay ripening, and then we focused on 3 treatments because these had the largest numbers of observations. And these were antitransparents, delayed pruning, and late source limitation. This is an example of a forest plot, which is the preliminary meta-analysis. So we have, for example, late defoliation, which is on treatment. We have our study details, and study ID, and some experimental conditions. For example, the first one is defoliation applied at pre-veraison on Ortrugo wine grapes from Italy, Magic, and in vintage 2017. And for each one of these observations, we have our TSS delay, the effect size, and the standard error. So what the forest plots actually pulls out an average effect, very funny again, and confidence interval. And you can see that based on this model, late defoliation can cause a delay of about, or an average delay of 1.04 bricks, and the confidence interval is higher than 0, so it's positive. What the forest plot does is actually fitting a random effects model where each study is can start from a different intercept. So it's allowing some random variation, obviously, between the studies. However, I'll go back to that. We can see in here that all these studies applied late defoliation, but the experimental conditions were very different. Pre-veraison versus mid-veraison, different varieties, different vintages, different sites, and so on. So we actually wanted to get into something called META-REGRESSION to understand the specific effects and the implications of environmental and experimental conditions on the outcome of these treatments. We did this using linear mixed models because that allows to fit vintage or site as random effects. because we cannot explain all the differences related to winter-herbicides, we can tell the model to actually allow them to randomly vary without describing the differences. This is, for example, a model for antitranspirants. We wanted to explain the ripening delay by a measure of the accuracy of the experiment, plus some environmental condition, plus experimental conditions, and these are the random terms. And just touch on some, a couple of key results and why they're important, we think, for industry. These are the significance of environmental and experimental factors for antitranspirants model. So no effect of environmental conditions, no effect of the precision of the study, but some experimental conditions were actually pretty significant. One of them was the type of antitranspirant spray you use, the active ingredient you use. This is where meta-analysis is pretty cool, because a couple of studies have looked at this into obviously 1 or 2 sites, but we can actually base the comparison of 1-p-menthin versus kaolin efficacy on a wide number of studies. So the model is telling us that when we are using 1-p-menthin, we are actually more likely to get larger ripening delay. Another thing that probably growers are very interested to know is when to spray these compounds to get the most and the largest ripening delay. So timing was again significant, and here we have predictions for different timing of applications. And we actually saw that whenever you apply antitranspirant twice, especially an early application plus a later application, we are getting better outcomes in terms of delayed ripening. So this also makes sense, and this is very important from a meta-analysis approach. You fit the model, but it has to be meaningful from a cultural perspective in this side. And obviously, we can think that this is due to the fact that we are repeating the application very close to when the sugar accumulation, the actual sugar accumulation starts. So there is where we are delayed ripening. And finally, we to just show where this can go, we created a little dashboard where growers can actually put in their different conditions at their experimental site, GDD's expected rainfall, average rainfall, and then when they want to pick the grapes and depth, and this little dashboard gives back a prediction of the ripening delay you will get by using different active compounds or different spraying stages. And this is what we're working on. So meta-analysis, pretty cool tool to review the efficacy of vineyard practices to delayed ripening. Some other nice outcomes, we saw that delayed ripening, delayed pruning is affected by environmental condition, and it's the only treatment. And this makes sense because this is the only phenology-based treatment. So we're pruning the grapes very early, and then a lot of things can happen during the season for this vine to catch up. We didn't see the same effect for antitranspirants and late source limitation, but obviously we are applying this much closer to the actual sugar accumulation. And we also saw that the most important factors remain yield and crop load. This again makes sense because these sink source relationships are those who are driving sugar accumulation. In general, so we wanted to look at the sugar flavor nexus in a bit of a more detailed and accurate way, and we combined different approaches to do this to both get a fundamental understanding and also understand implications for industry. The first bit was a single bearer approach. We prevented the confounding effect of asynchronicity, and we started and we were able to better characterize the relationship between metabolites and sugars in developing berries. In the second bit, we used field trials, and this led to both a fundamental and applied outcome. We saw that there is a certain decoupling between sugar accumulation and aroma development, and this we saw it because we varied the sugar accumulation, but on the other hand, the sugar aroma development seemed to happen on its own way, so we can actually shift sugar accumulation to an earlier and later period to target different aroma composition. We also reviewed techniques to delay sugar accumulation because we saw that this is very important and their efficacy under different environments and growing conditions. And I really want to acknowledge the supervisory panel here, Chris, Kerry, Wilkins, Kerry, and Nick. It's been amazing to work with them. Wine Australia for the supplementary scholarship, all the people in the wine science group, Isagao Winery and the meta-analysis gang, and of course, all the support from the training center and the involved industry partners. And I'll take any questions, feedbacks. This will help me to write my thesis, so please.
Thank you very much, Pietro. If anybody has any questions, if you can type them into the Q&A box, and we've already got one. Okay, we've just got a comment from Vlad, a great talk and research effort. Well done to you and your supervisors. So does anybody have any questions for Pietro?
I love that type of question.
That's the easiest
There's one in the
question.
chat. There's one up in the chat.
Oh yeah.
Something.
Okay. Another one. Excellent talk. I would love a question because it was a great talk, but I don't have any direct questions for Pietro. So fantastic if somebody online can ask a question. Otherwise, we all get an early lunch.
Exactly.
Right? Okay. Easy. If there are no questions, I'd like to thank Pietro very much for a really interesting talk and a really good presentation, and wish you all the best for finishing off your thesis.
Thank
Your
you.
return to Italy.
And if you have any question, just email me or something. There's always another way.
Perfect.
Easy. Thanks,
Okay.
Rebecca.
Thanks, everybody.
Thanks, everybody.
Okay. Sorry.